We'll talk about cell cycle regulation today. Okay. Cell division was almost completed. A little bit part might be remaining, like difference between the mitosis and meiosis. And I think you will be able to do this that part by yourself. The significance of meiosis, significance of mitosis, find out the answers for these types of questions, and you accumulate those questions related to cell division questions, whatever you find in previous year's questions. You collect those questions and write down in a particular place, in a diary or in a notebook, and try to answer those questions. If you don't find any answer of any of them, those questions you might discuss here. But before that, you should gather those questions from everywhere, from the internet, from different types of questions that are asked in different exams. Not only your exam, even in Indian Forest Service, Indian Administrative Service, or any other master's degree exam or whatever entrance exams are there. You collect those questions and clear your doubts over here. That part is your own, your job. Okay. Now, so I'm going to proceed to the next level, that is cell cycle. I think you are able to see the screen. Let me switch on someone. Everybody's mic is off. Swati Sharma, can you see the screen?
Yes, sir.
Okay. Yeah. You'll be there as monitor. So here is the, if I go by this one, so a number of books are there in which in cytological cell biology books, cell and molecular biology books, you will get this thing, the cell cycle. What is a cell cycle? You know about this cycle, that the cell cycle is a time-bound different events that occur during the life of a cell, and in the definition, what is mentioned is this, that if you start from a particular place, say from prophase, and if you return back again to the same phase, the sequence of events that occur between these two phases, prophase to prophase, is one cell cycle. Okay, what are the sequence of events that occur? First is the divisional phases, the prophase, metaphase, anaphase, telophase. And after that, when the cell has divided into two cells, it undergoes a preparation, that is gap 1 phase, and then synthesis of the DNA, that is duplication of the DNA, replication, you can call it, and then again preparation for cell division. So that is called gap 2. And each of these phases are highly regulated, whether the cell will enter from G1 to S or not, or S to G2 or not, whether the cell is prepared for division from G2 to M or not. This has to be checked. So there are checkpoints at each of these junctures. Okay, at each of the junctures, there is a checkpoint. Particularly, you may say that from mitosis to G1, the checkpoint is very, not that strong checkpoint, but G1 to S is a very, very strong checkpoint. S itself is a very strong checkpoint. From S to G2 is a very minor checkpoint, but G2 to M is a very strong checkpoint. And the next checkpoint is near the metaphase and anaphase stage. In the anaphase stage, particularly when the chromosomes are going to be separated. So the cell has to assure that the chromosomes are perfectly separated. So that is also a very important checkpoint.
And there is a stage called G0 stage, which is a resting stage, you can say. Some of these cells, they do not enter into the S phase immediately, and they may undergo a sort of if they are differentiated, they might be in the G0 stage for the whole life, you can call the neuron cells and the muscle cells. But there are other cells like in our body. In case of plant cells, G0 stage will be all those differentiated cells that never go de-differentiation. Like you can say the xylem and phloem that is once being formed, they remain as it is. So that enter into G0 stage and they never undergo division further after maturity. Even the cork cells, you can say, that is the periderm, but the cork cambium or the vascular cambium, that enters into a G0 stage for a certain period of time, but when secondary growth begins, they again return back to mitosis. So they have to come back to G1 stage again from G0 stage. So in case of plants, that is the case. In case of human being, you can say that the muscle and the neuron cells, they enter G0 stage permanently, but there are other tissues like the liver cell or kidney cells. The kidney and liver, they are made up of cells which undergo sort of a rest for a certain period of time. But if damage occurs, if you excise out certain part of your liver, it might grow into a full liver, a normal liver. So it has a potency to divide and reform. So these kind of cells enter into G0 stage for a certain period of time, and they might return into G1 and S phase again. But there are others which do not enter into G0 stage in that way. For example, the skin cells, you can say, the skin cells and the bone marrow pluripotent stem cells. These cells, they have a continuous mode of cell division. They do not enter into G0 stage. So they directly enter into the G1 and S phase and G2 phase for next round of cell division. So as far as the definition is concerned, this goes like this. The cell cycle of individual cell considered in the perspective of the timed sequence of morphological and biochemical events occurring in it during the period between a particular recognizable stage to the next similar stage. This was a definition given by Watson in 1976. You see here, between a particular stage, between a particular recognizable stage, this can be any stage, like the prophase, metaphase, anaphase, telophase, or G1 or S or G2, whatever. Wherever from you start, when you return back to the similar stage, the sequence of events that occur between these two stages is one cell cycle for an individual cell. That is the definition which means to say. So the cell cycle can be divided into two parts, as you know, mitosis and interphase. The interphase was like this: gap 1 or G1 phase, synthesis phase, and gap 2 phase that we already talked about, and the G0 stage that we were saying. So now, what is the regulation that undergoes during these different phases? And why is this regulation is there? Why the regulation is there, you can understand that if the cell division is not controlled in that way for a particular cell, in a particular tissue, the behavior of that tissue or the cell will not be exactly the same as it is desired from that particular cell. A cell from a kidney cannot behave similar to that a cell of a liver or a brain or muscle cell or a different tissue cell. So each of these cells have a different nature. The gene expression pattern is also different.
there are housekeeping genes, about 70% of the housekeeping genes which are common to all cells, and they are constitutively expressed. But there are 30% genes which are differentially expressed in those particular tissues only. The genes required for photosynthesis will be expressed only in mesophyll, in chlorophyll containing cells, in chloroplast containing cells, but they will not be expressed in cells that those don't possess chloroplasts or chlorophylls like this. So those genes will be there. In the chromosome, the gene will be there, but its expression will not occur. That is the thing. The number of genes does not decrease cell to cell. It remains the same. The number of chromosomes also remains the same in all the cells of a particular individual. Now, coming to the cell cycle is a highly regulated process. If a cell which normally has to remain in G0 phase continues to divide, it may cause cancer. Thus, every stage of the cell is under the control of various regulatory proteins. Now, here we will talk about two principal proteins that are called cyclins and cyclin-dependent kinases, CDKs, you can say. These two are together called maturation promoting factors. And they were discovered in the sense where there is a frog, marine frog called Xenopus laevis. Okay, when scientists took the oocyte of mature oocyte of Xenopus laevis, and they extracted and they took the nuclear extract of this oocyte, mature oocyte from Xenopus laevis, and they put that nuclear extract into an immature cell. The immature cell immediately become mature. Okay, are you getting this? When you are taking nuclear extract of a mature oocyte cell from Xenopus laevis, and you are adding those extract into an immature cell, the immature cell is immediately transformed into a mature cell. So what is making this transformation to occur? That particular factor is present in the nuclear extract, definitely, and that was called maturation promoting factor because it promotes the maturation. Later on, it was known that this maturation promoting factors actually comprises of two different proteins called cyclins and cyclin-dependent kinases. Because these are the kinases. Kinases are the proteins that add a phosphate to a particular substrate. So kinases add phosphate, and phosphatases remove phosphate from that particular substrate. So, and these kinases, they are dependent on cyclin. Until and unless they bind with cyclin proteins, one or the other type of cyclin proteins, they will not get activated. So they are dependent on cyclins. That's why they're called cyclin-dependent kinases. So cyclins, and why the name cyclin is there? Because these proteins are cyclically formed and degraded in the system. The name cyclin is there because they appear in the cell at certain point of time, and when the function is over, they get degraded by mechanisms that are that you might be knowing, that our cell poses a recycling mechanism. The proteins that have completed their function, they undergo processing and they are converted into the raw material like the amino acids by a degradation process, a proteolytic degradation process that is based on a particular machinery that is called proteasome. So proteasome is an instrument. When a protein passes through that proteasome, it gets degraded into its finally into its amino acid forms, and those amino acids can be used for protein synthesis in the next round of protein translation. So not necessarily those amino acids will be used for the same formation of the same protein. They may be used in other synthesis of other proteins also. So because they are present in the cellular environment, and there is no mechanism to separate those amino acids, that these have been produced by this particular protein, so they will be used for that formation of that only. That is not necessary for the cell. to distinguish. So cyclins are, that's why the cyclins are named like that, because they are formed once and they, after the function, they are degraded by proteasomal mechanism. You should go and see how proteasomal degradation occurs in Google or in your book. Write down proteasomal degradation. You need to see that how it occurs. It is a drum-like structure. You can get this in cell biology books, in biochemistry books. And proteasome are a drum-like multi-protein subunit structures. A particular protein which has to undergo degradation has to be labeled. That protein which has to go to the proteasome, that must be labeled or tagged. So how that protein will identify and how the system will identify that this protein is going to be degraded through proteasome, that protein has to be labeled, and that process is called ubiquitinization. Ubiquitins are proteins that are added, that are tagged, you can say, on those proteins which are to be sent to the proteasome. So many such ubiquitin proteins are tagged one after another, and that protein is now ready for going to proteasome for degradation, and the process is called ubiquitinization, followed by proteasomal degradation. So cyclin proteins undergo like this. So what are maturation promoting factors? Maturation promoting factors are proteins that convert an immature cell into a mature cell, and they comprises of two main principal proteins that are called cyclins and cyclin-dependent kinases. Okay? These cyclin-dependent kinases, in some other books, you might find they have been written in CDC2. Here you can see this one, CDC2, CDC, cell division cyclins. This nomenclature was an earlier nomenclature based on Saccharomyces cerevisiae on the basis of studies on yeasts. They were said to be like this. Cell division cycle proteins, they are called. Cell division cycle, CDC proteins. Earlier, these CDK proteins were called CDC proteins based on their studies in yeasts. But later on, they were termed as CDK. So you might get this kind of designation like CDC2, CDC28. They mean CDK28, CDK2, like this. In human, we call it CDK. In yeast, we call it CDC. But alternatively, you can call CDC as CDK in yeast also. CDK is more acceptable. Okay, and it actually is the term. Nomenclaturally, it is more perfect because it is a kinase. But here from CDC, you are not clear what is the function of this protein, cell division cycle protein. The cyclin and CDCs are not very different. It seems they are same. But actually, CDC is same to CDK. So this I'm saying, when you are reading a book, you might face this CDC term, and CDC are basically CDKs. Here we can omit this part. Based on their function, CDKs are perceived to be as HR. If you consider this as I'm skipping this area now, and to make this thing simple, let us now see in which particular stage of the cell division, which kind of CDC and cyclin are expressed. So there are cell cycle phases, G1. G1 can be divided into two phases, early and late. Okay. In the early G1, you see cyclin D. DEAB. There are four cyclin, they will come one after another. DEAB. Or you cannot start from AB. A is the protein which is expressed during S phase, cyclin A. So S comes later. So first comes the G1 phase. In G1 early phase, cyclin D is expressed, and along with cyclin D, either CDK4 or CDK6 will be operative. In G1 late, that means where G1 is entering into the S phase, that particular transition phase, at that juncture, cyclin E is expressed. And that function is controlled by cyclin E and CDK2. So you remember this. S phase is controlled by cyclin A and CDK2. and M phase is controlled by cyclin B, or you can say G2M phase. G2M phase is controlled by cyclin B, CDC2 or CDK1. Okay. किसके प्रॉब्लम है? स्वाति शर्मा. हाँ, आवाज तो इधर से जा ही रहा, फिर उधर से आने से प्रॉब्लम है. So we were seeing this thing. So you remember this particular table, that in G1 early cyclin B, in G1 late cyclin E, in S phase cyclin A, M phase cyclin B are expressed, and along with those, which particular CDKs are expressed. Just a minute. Hello. So this thing has to be kept in mind. Otherwise, you cannot, you'll find difficulty in answering. In that case, if you are forgetting the names of these D, E, A, B, and these things like CDK4, and if you are not able to remember this, you can do that. You at least you know there is a particular cyclin which is expressed in G1. There is a particular cyclin which is expressed in S. And accordingly, there are particular CDKs expressed in those phases. If we're not able to remember the names of these, this might happen, but you don't forget the concept that a particular cyclin has to be activated. The name might differ, but it has to be activated, and that has to bind with the particular CDK, and that CDK will execute a particular kind of a function. Okay, so what happens? How do cyclin and CDK interact? How do they function? When they are independently present, they are inactive. But when a cyclin associates with a CDK, any of those pairs, I mean cyclin D to CDK4 or 6 cyclin E to CDK2, these are the pairs, you can say. So when a cyclin binds with a CDK, it becomes partially active. But it becomes fully active when the 161st threonine amino acid of CDK becomes phosphorylated. Okay, this is the active form of this doublet, you can say, heterodimer, that has to be associated. So how this is phosphorylated? There are 3 steps for that. There are 3 proteins associated, V1, CAK, and CDC25. You just remember these things. V1 and CAK are 2 kinases, and CDC25 is a phosphatase. Okay, V1 and CAK are 2 kinases. V1 adds a phosphate, phosphorylates the tyrosine, 15th tyrosine residue of CDK. Okay, first. And then the cyclin activated kinase, this one, CAK, cyclin, this is called CAK activated kinases. CDK activating kinases rather, not cyclin activating kinase, CDK activating kinase. I was searching for this. And V1 is tyrosine kinase, protein tyrosine kinase. It phosphorylates the tyrosine residue. And CDK activating kinase, it phosphorylates the threonine, 161st threonine residue. Okay. So in the next, I mean, there are 2 rounds of phosphorylation. One is at 15th position in the tyrosine by V1, and the other is at 161st position in the threonine amino acid. So when these 2 get phosphorylated, this dimer is still inactive. But now when now CDC25 removes the first phosphorylation, that is the tyrosine phosphorylation now. Okay, and only the P61 phosphorylation remains. This is the active form of this cyclin CDK complex.
Okay, so this is how cyclin CDK complex is activated in each of these cases. Cyclin D with CDK4 or 6, cyclin E with CDK2. So this is the basic, and now let us see at every places wherever a cyclin is involved, there is a counterpart CDK for that, and they undergo activation in this mechanism. Now, there are several checkpoints in the cell cycle. First one is the G1S transition checkpoint. Okay, then there is S phase checkpoint. Then there is G2M transition checkpoint. And finally, there is a Anaphase, metaphase Anaphase checkpoint that we will discuss here. Now let me show you this diagram. With the help of this diagram, I will tell you how things are happening. Okay, let me ask whether you are there or this is actually not an interactive platform. Abhira Mandal?
Yes, sir. Yes, sir.
You don't know. Sudeep. সুজান আমি 5 জনকে অন রাখলাম, ঠিক আছে, বাকি পরে আমরা 5 জনকে অন করব। তোমরা আমাকে মানে সাবজেক্টে শুনতে পাচ্ছ তো?
হ্যাঁ স্যার।
এতক্ষণ যেটা আমি মানে এইটাতে না বুঝতে অসুবিধা হয়। আচ্ছা, যদি কারোর প্রবলেম বাড়িতে আওয়াজ হচ্ছে তো একটু আমাকে বলবে যে আমারটা অফ করে দিন। তো this is the G1 phase, okay, and this is G0 phase. You don't consider G0 phase in this particular when you are talking about regulation. G0 is a resting stage for certain kind of cells. When they enter into that cell, they remain, as I told, that differs. The timing of the G0 stage differs. It may be from 0 hour to the whole life the cell is there in the individual, okay. So how is G1 phase regulated? G1 phase is regulated by a protein complex that is called RBE2F complex. RB is retinoblastoma. This is oncogene retinoblastoma protein, and when it binds with the elongation factor to E2F, which is required in translation, the cell cannot enter. In this form, the cell cannot enter in the S phase. Okay, when RB is associated with E2F, the cell cannot enter into the S phase. RB has to be set free. Okay, RB has to be relieved, or E2F has to be set free. Rather, better to say that these two complexes has to be broken down, and that will happen by the phosphorylation of the retinoblastoma protein. There are two, this is a hypophosphorylated state and this is hyperphosphorylated state. So by two steps, there are two phosphorylation steps by which this retinoblastoma is dissociated from E2F. And when E2F is free, the cell can enter into the S phase. So that has to be executed. And how that is being done? Retinoblastoma E2F is first phosphorylated by the CDK4 or 6 CDK. And that is being controlled by cyclin D, as we mentioned earlier. Cyclin D and CDK4 or 6, this is the pair that acts in the early G1 phase. So it first phosphorylates, it hypophosphorylates the retinoblastoma, and that's why some amount of E2F is released, but that is not enough. And in the further phosphorylation of retinoblastoma is needed, and that occurs. When that occurs, the E2F is completely ready to function, and the cell will enter into the S phase now. But these two proteins, the later part is regulated by cyclin E.
Cement আছে ওইখানে, Yes.
Cyclin E and CDK2. Okay, this acts in the later part of this G1 S-phase transition, you can say, and they are in turn under the control of a protein called P21. P21, it is a kind of oncogene, suppressor gene, you can say. This is a suppressor gene for cell division, and this symbol, I mean, there is a bar and like this. If there is an arrow, that means it promotes the process, and if this is blunt like this, that means it inhibits this particular process. So P21 is an inhibitor of cyclin D CDK4/6 complex. It is an inhibitor of cyclin E CDK2 complex. Okay, P21, and P21 is again controlled by another protein, suppressor protein P16 that has been not written here, or you can directly go to P53. P53 is a very, very, very important protein. It has been seen that in about 50% of the cancers, the P53 gene is involved in one or the other way. P53 gene gets mutated, the cell might become cancerous. And that's why this P53 has been called guardian of the genome. It is the guardian of the genome, P53. So if you search P53 in internet, in Google or in NCBI, you will find cancer-related papers. More than 50% of the papers will be linked with P53 gene. So very crucial. Once upon a time, when cancer biology is still a very burning topic in research, so it has not lost its its shine. P53 still is a burning topic. However, there are other things that have come, like the miRNA, siRNA, those kind of things, and regulation, stem cell biology. Those have taken most of the part of the research now. But many scientists, they still, cancer biology is the burning topic in research till now. So P53 regulates P21. It says P21 go and inhibit cyclin E, cyclin D, and don't allow the cell to enter into S phase. Why? Why does this do so? Why P53 will tell the P21 to inhibit these cyclins? Because if the chromosome of this cell, that is the DNA, if the DNA of this cell has undergone certain kind of mutation or damage during this cell division process, then that damage has to be repaired before the cell enters into the next round of cell division. That has to be seen whether the chromosome is intact or there is any problem, and if there is any problem, that has to be solved, isn't it? Without correcting those problems, the cell cannot enter into the next phase. That is the synthesis phase. Synthesis phase will synthesize the same thing. That is, if it is an error, it will also synthesize an error DNA. So the error has to be repaired before this, before this S phase, during this G1 phase juncture, when the cell is going to enter into the S phase. Before that, so that's why if there is an error in the DNA, if there is a damage in the DNA, P53 will inhibit the process with the help of P21. Okay, so that's why they are called suppressor genes. P53, P21, these kind of genes are called suppressor because they suppress the cell division. Okay, and P53 is under the, I will not take you to that level. Now I'm telling the story, but you might skip this part. You simply may write that P53 is a guardian of genome, which regulates the function of P21. If the DNA is damaged, the cell cannot be allowed to enter into the S phase, and so P21 inhibits the function of cyclin D and cyclin E, so that the cell does not enter into the S phase. You can write up to this, not more than that. But a little bit of story is the function of P53 is regulated by another protein called MDM2. So MDM2 binds with P53. Until MDM2 is bound with P53, P53 is inactive. It will go to proteasomal degradation. It will not be participate in this process, and this will happen only when the cell is okay. I mean, there is no damage in the DNA. But if there is damage in the DNA, P53 will be released from MDM2. It will get phosphorylated, and it will become active. Okay, if the DNA damage has occurred, P53 will get phosphorylated and will become active. It will induce P21, and it will tell P21 to inhibit these processes, these proteins, Cyclin-D CDK4 and Cyclin-E CDK2 complexes. And the cell will be arrested for the time being in the G1 phase. And the repair mechanisms, P53 is also involved with the SOS system, DNA repair system. You might see there, P53 save our soul. That is called when the system faces a situation when the DNA damage is very severe and all the repair genes has to be switched on, then save our soul signal is induced. And that is also under the control of P53. Okay, that means P53 is very, very crucial enzyme, isn't it? Anyway, so now, how I will not say how P53 is directly, I mean, it controls the DNA repair. When the time will come, we will talk about that also. So now the cell has this CDKs, Cyclin-D, CDK4, and Cyclin-E, CDK2, they have done their function. Now the cell has entered into S phase. Now coming to the next phase of regulation. Okay. So what is happening during this phase, S phase? The cell has to ensure, remember here, this is not a simple phase. The cell has to ensure that DNA replication occurs only once. ঠিক আছে? বাংলায় বলছি যে সেল ঠিক করবে যে রেপ্লিকেশনটা যাতে একবারের বেশি না হয়, না হলে কি হবে, একটা সেলে যতগুলো ক্রোমোজোম আছে সেটা মানে আই মিন প্লয়েডি লেভেল ইনক্রিজ করে যাবে, isn't it? এটা গন্ডগোল হয়ে যাবে। যদি একবার অ্যাপ্লিকেশন হয়ে গেছে আরেকবার হয়ে গেল, এই ব্যাপারটা যদি কন্ট্রোল না থাকে, তাহলে যখন আমরা সেল ডিভিশনের এই ফেজেতে যাচ্ছি তখন ক্রোমোজোম গুলোর কি অবস্থা হবে সেটা বুঝতে পারছো। যে ওই এক একটা ক্রোমোজোমের যদি তাহলে ক্রোমেটিড দুটো করে থাকে, তাহলে এক একটা ক্রোমোজোমের ক্রোমেটিডের সংখ্যা বেড়ে যাবে, তাই না? সেটা হতে পারে না, সেটা হতে দেওয়া হবে না, সেটা রেগুলেশন। This is the regulation that is working here, that the cell will ensure that the DNA replication occurs only once. তো কি হয় এইখানে at the beginning when the cell is entering to the S phase, এখানে কেউ যেটা origin of replication থেকে replication শুরু হয়, as you know, this is a big chromosome and there are so many origin of replications. At each of those sites, ORC, origin of recognition complex site, ORI site, there are some proteins that are binding there and they are inhibiting, they're binding there unless they dissociate from that place. Replication will not start. Okay, and that's occur when this cyclin E becomes E2F becomes free, and it induces cyclin A protein over here. Cyclin A, this one, Cyclin A CDK2 proteins here. Okay, now the function is of Cyclin A CDK2. What they do, they remove the inhibitors. These are the inhibitors, CDT1, CDC6. These are the inhibitors, and there is another inhibitor that is called P27 KIP1, KIP1. Three inhibitors mainly. Okay, there are three inhibitors mainly. P27 KIP1, CDT1, CDC6. So those have to be removed from this place. This is the origin of replication complex, and MCM hexamers, MCM6. This means MCM2 to 7. There are 6 such MCMs. Those are called heterohexamers. They will come and bind at this place. Okay, these will be removed and MCM will come and bind at this place. These MCMs are the starters. I mean, when they will bind, replication will start. And when the replication fork will proceed, the helicase enzyme will come and bind over here, and the replication will proceed on two directions further. What will happen as soon as the DNA has doubled, it will again be bound by these CDT1 and CDC6. The origin of replications will be bound by these proteins as soon as the replication will be completed, so that the cell cannot enter into the next round of replication. To ensure or to inhibit the further replication, once the DNA has replicated, the CDC6 and CDT1 and the P27 KEAP1 protein, they will start their function and they will inhibit the next round of replication. So this is the replication-related regulation that occurs during this S phase. Okay, I think you are getting these things. Now comes the G1 phase. G1 phase is preparation for the entry into the divisional phase. So this phase is controlled by, particularly the G2 to M transition, this phase is controlled by cyclin B and CDK1. You remember three things happen here by the phosphorylation activity of cyclin B, CDK1. What they do? Basically, all these cyclins, they phosphorylate. You can see everywhere there is a phosphorylation process. This CDK4/6 phosphorylates retinoblastoma. Again, CDK2 cyclin E, they phosphorylate retinoblastoma further. And then it waves, so that's why it is released. Here also, CDK2 and cyclin A, they will phosphorylate this CDT1 and CDC6 so that they are released. Here you can see there is a phosphate attached to this blue protein, which is CDC6. So phosphorylation mechanism is everywhere. So here also CDK1 and cyclin B, they phosphorylate three different proteins: H1 histone, nuclear lamine, and cytoplasmic proteins, because these are required in the next round. What happens if H1 histone is phosphorylated? The chromosome will become thicker. The chromosome will start condensing. It will enter into the metaphasic stage of the chromosome. You know that from prophase, the chromosome starts become thickening. The chromosome becomes fattier, I mean more compact and thicker, and that is because of the phosphorylation of the H1 histone. Okay, and then শুভদীপের হচ্ছে নাকি দুবার শুনতে হ্যালো না শুভদীপের হ্যালো না সুজানি না হ্যালো আবির একটা ছিল Okay, I have withdrawn some other people. Now, about say, अरे क्यों कि हेडफोन हेलो हेलो Hello. Smita type problems. Hello. Smita. Anyway, so we're in a very interesting place. You can see because of these three phosphorylation, the cell becomes ready to enter into the divisional phases. You just see the changes. When H1 is phosphorylated, H1 histone, the chromosome condensation occurs. Okay. When nuclear lamine is phosphorylated, the nuclear membrane is degraded. It starts, the next step is what? In prophase, we see that the nuclear membrane is disappearing, and that is started by the phosphorylation of nuclear lamine proteins. Then cytoplasmic proteins, that means the actin proteins and the other proteins, the band 3, the glycophorins, those are the cytoplasmic proteins, and they get phosphorylated, particularly the actin protein. And then what happens? Because during cell division, the cell morphology changes. The cell membrane becomes elongated, and then a constriction occurs near the equatorial zone in case of animal cell particularly. In plants also, the cell increases in size. So there are a lot of changes in the cytoplasm, in the structure of the cell, in the cytoskeleton, you can say. So the cytoskeletal proteins, they are phosphorylated so that those changes can be accommodated in the system. So this thing start happening during this G to M transition. Okay, you remember this? And afterward, when the next round of control is near this metaphase and anaphase stage. They're very crucial control over there. In our last class, we were seeing that the chromosomes have to be brought into the equatorial region, so that the separation occurs in equal numbers on both the poles. So that has to be assured. Who does that? How the chromosome system assures that the chromosome, the spindle fibers have bound to the centromeres, and until and unless all centromeres are bound by a particular spindle fibers, this next phase, that is the anaphasic separation, will not begin. Until and unless all the chromosomes are brought to the middle of the cell, that is the equatorial zone, anaphasic separation will not begin. So this is under the control of spindle assembly checkpoint, SAC, a very crucial checkpoint, okay? Remember, spindle assembly checkpoint, SAC. And that is under the control of a protein. SAC is under the control of a protein that is called MAT2. So MAT2 is basically a wait signal. Wait means the cell has to wait until the chromosome have come to the middle of the cell for separation to begin. And equal number of chromosome has to be divided into two and has to go to two poles for the formation of two different cells. Here, this is a very crucial area, a crucial place where chromosomes are getting separated, and they have to get separated in equal. They have to get the equal number of chromosomes, equal number of genes, and all those things, isn't it? So this regulation is very crucial. All regulations are crucial. This is also very, very crucial place. Okay, so the wait signal, it is a function of the kinetochore-associated protein MAT2. It controls the spindle assembly checkpoint so that all the microtubules, all the spindle microtubules, not all, I mean all the centromeres rather, in other words, I should say, all the centromeres or the kinetochores, they have bound with a particular spindle microtubule or not. Until and unless that happens, the cell, it will not be allowed to enter into the anaphasic separation. So once that has happened, those proteins which we talked about, those are kinesin 7, kinesin 13, and cytoplasmic dyneins, I think you remember. If you don't remember, it will be difficult. I have told you that the cell biology, molecular biology is such a thing that you cannot simply memorize and you just mug up and go and bomb it there in your copy, in examination script. You can do that. That will not be, that will not last longer. Just for carrying marks, you can do that. But for understanding, you have to know the story one after another, and each of this is linked. So now, next thing is this anaphase promoting complex. Then metaphase has occurred, this chromosomes have been brought to the middle of the cell. Now separation has to take place. And that is being controlled by anaphase promoting complex / cyclosome, APCC checkpoint, that is called. What is that checkpoint? This APCC checkpoint, it is associated like this. Now, in the middle of the chromosome, middle of the cell, the chromosome is like this. There are two chromatids and there are two centromeres. Here it has been that there are two centromeres. Here also there are two centromeres. They're not exactly visible, but a little while later they will become visible like this. And so there are two centromeres. Centromeres have been formed during the S phase when the DNA was replicating from one chromatid to two chromatid in a chromosome. The centromeres also got replicated that time, but they were not visible. The visibility occurs during the metaphase. Okay. So, but these chromatids are bound by a protein that is called cohesin. cohesin proteins, they actually tag these chromatids like this. Okay, they're not free. Okay, they are being, so this has to be removed. That this binding has to be overcome. Okay, these cohesin proteins has to be removed, and that is being done by this APCC complex, which is under the control of CDC20. CDC20 is again under the control of MAT2. As we said, that the MAT2 is the weight point, is a weight checkpoint until chromosomes are brought to the middle. It will allow the CDC20 to promote APCC complex. It will activate this one, APCC. CDC20 will activate APCC. Now, APCC is basically you can say it is a ligase. It is a ligase which ligates ubiquitin proteins. Ubiquitin. I told you about the ubiquitinization and proteasomal degradation. So this APCC it requires a ligase protein. Ubiquitination is a ligation process. Ubiquitin has to be tagged with the particular protein for degradation, and that is the ligation process. An enzyme called ligase ligates the ubiquitin moieties to a particular protein so that that protein can go to the proteasome for degradation. So here the same thing is happening. This APCC is basically a ligase. It ligates ubiquitin molecules to separin residue. There is a complex protein, securin and separin complex, and this securin has to be removed. Separin has to be removed. This will be securin, not separin. This has been written both places. It has been written separin. This one is securin. This one is separin. So securin is tagged with ubiquitin proteins so that it goes for degradation via proteasomal degradation. So now separin is free. When separin is free from securin, it will separate. It will actually digest these cohesin proteins, and these chromatids will get free now to move to the two poles. Okay, so now the thing will happen. These spindle fibers that have attached to these centrioles, they will take these two chromatids to the two poles. Okay, and the job of this APCC has been completed. A little bit of that is that it also induces a CDH1 protein, and that clips the cyclin B protein. As you know, cyclins are also cyclic proteins. They are degraded proteasomally after the function is over. So here, the cyclin B, as soon as the cell division has, the anaphase has entered, the cyclin B has to be degraded. And that is being induced by CDH1. CDH1 is also activated by APCC system. So APCC does two things. It degrades the cohesin proteins, which actually hold the two chromatids together. When cohesins are removed, the chromatids are ready to go to the two poles. Anaphase has started, and at that time the function of cyclin B is over, and that has to be degraded, and that is being done by CDH1. And that again is induced by APCC APCC cyclosome protein. That's all about. I think in this 1 hour lecture, I told you everything, almost everything, what you require to know about cell division at this level. There are